to this video lecture, and in this video lecture I'll be solving previous year's Neet questions and discussing the answers. And we are doing this for the chapter Excretory Products and their Elimination, as you can see on your screen, and this is part 3 of the series. Okay, so let's begin. Okay, so before I begin, let me change the pen. Okay, so question number 1, which of the following would help in prevention of diuresis? Okay, which of the following would help in prevention of diuresis? So you have your options A, B, C and D, and this appeared in Neet 2020, which is quite recent. Okay, so I would suggest you pause the video, think about the answer, and then come back and we can watch the discussion. So let's go through our options. More water reabsorption due to undersecretion of ADH. Okay. B, reabsorption of sodium and water from renal tubules due to aldosterone. C, atrial natriuretic factor causes vasoconstriction. D, decrease in secretion of renin by JG cells. Okay, now let's read the question. Would help in prevention of diuresis. Now what is diuresis? Right, what is the meaning of diuresis? Now diuresis means renal output of water. Okay, the renal output of water. This is the meaning of diuresis. Okay, so now we have to find out what would help in preventing that. So let's look at our first option, A, more water reabsorption due to undersecretion of ADH. Now what is ADH? In case you didn't know, it's anti-diuretic hormone. In case you didn't know, ADH is anti-diuretic hormone, which you will come across when you study chemical coordination. Okay, and as you can see from the name itself, it's anti-diuretic hormone, right? So now what is the job of ADH? Ideally, what is the job of ADH? Now when ADH is secreted, okay, it is secreted from the posterior pituitary gland, okay, and it attaches to the receptors, okay, in the collection tubule and in the later distal part of the tubule and the collection tubule. It is attached over there. There are some receptors present over there. Now the job of ADH is to produce or form water channels. Okay, it's called aquaporin water channels. Okay, now when these water channels are formed, they're formed on the luminal side. Okay, so if you have your tubule, your cell, this is your cell, tubular cell, let's call them tubular cell, and this is the interstitial fluid side, and this is the tubular, the filtrate side. Okay, now there are some V2 receptors. Let's take another color. V2 receptors over here. Okay, and ADH comes and attaches itself to the V2 receptors. Now series of chemical reactions happen. Okay, so CAMP gets activated, and then it activates protein kinases, which we will not go into the detail. Okay, these are cellular mechanisms, we will not go into that. So now what happens as a result of that? As a result of that, aquaporin channels are formed here. Okay, on the luminal side. Okay, aquaporin channels are formed here. They're called AQP2. Okay, aquaporin channels are formed here, AQP2. Now these water channels, okay, these are water channels. They are highly permeable to water. They allow water to enter. Okay, on the other side, the basolateral side, there are other channels called AQP3. Let me just erase this, take another color. So you can differentiate. Okay. On the other side, there are AQP3 aquaporin, okay, AQP3 and AQP4 water channels are present, which allow the exit of the water molecules and into the interstitial fluid, which is our job, correct? This is when normal ADH is there, present there, and water reabsorption needs to happen. If under secretion of ADH is happening, means what? Water reabsorption will not happen, obviously, right? So when under secretion of ADH, water reabsorption will not be more, it will be less. Okay, so this statement is incorrect. It will not prevent diuresis. More water reabsorption due to under secretion of ADH is an incorrect statement. Okay, there will be less reabsorption due to under secretion of ADH. Less water reabsorption due to under secretion of ADH. Okay, so clearly A is incorrect. Alright. B. Reabsorption of sodium and water from renal tubules due to aldosterone. So aldosterone, yes, does play an important role in the reabsorption of sodium. Okay, reabsorption of sodium from the DCT, the later part of DCT and the cortical collecting tubules. Okay, that region is very important. There are some cells called principal cells. Okay, there are some cells, special cells called principal cell in the later part of DCT. If you watched my previous lecture, you would know what I'm talking about. There are some cells called principal cells, and aldosterone act on those cells and they allow the reabsorption of sodium. Although that region is not permeable to water, only like not by osmosis, but only when ADH attaches there. Okay, so that region is not permeable to water as such that when sodium moves, water will also move. Doesn't happen like it works in PCT, the proximal side, okay, or the loop of Henle side. No, that region, the later part of DCT and the cortical collecting tubules are relatively impermeable to water. Okay, they don't move out as per osmosis, but they move out only when ADH attaches over there, like I've already explained to you over here. Okay, this part, this is the mechanism. So reabsorption of sodium and water from renal tubules due to aldosterone, yes, that will prevent diuresis, right? That will prevent the renal output of water. So this is the right answer. We look at the other options. C. Atrial natriuretic factor causes vasoconstriction. Now, it's not actually atrial natriuretic factors, atrial natriuretic peptide. That's how it's denoted in medical textbooks. But yeah, if NCRT is writing it as factor, just follow that. But ideally, it is ANP. You won't see it as ANF, really. Okay, so natriuretic is what? What is the meaning of natriuretic? It is the output of sodium ions, renal output of sodium ions. I hope you can see. I'll choose another color. Oh, this was. Let's choose this one then. Okay, this is a very not a very good place to write. But anyway, sodium ions. Okay. So atrial natriuretic factor causes vasoconstriction. Clearly, the statement is incorrect. It does not cause vasoconstriction. What does ANP cause? ANP causes vasodilation. Okay, ANP causes vasodilation. Okay, and this ANP is released when there is high blood pressure or volume. Sorry. Okay, when there is high blood pressure or volume, ANP is secreted. Okay, it's released. Okay, and then this acts and causes vasodilation, thus increasing the blood flow. Okay, the blood volume, the blood pressure. I'm sorry, the decreasing the blood pressure, the blood volume. I'm sorry, it's the opposite. So it's causing vasodilation to decrease the blood pressure or the blood volume. So it's secreted when there is high blood pressure or blood volume. Okay, and then what it does is causes vasodilation, and therefore that blood volume or the blood pressure decreases. Okay, so C is an incorrect statement. It will not prevent diuresis. D, decrease in secretion of renin by JG cells. Again, this is an incorrect statement. Okay, now whenever you need to prevent diuresis, for diuresis to occur, ANF will cause vasodilation. Okay, it will cause vasodilation. It's the opposite effect, right? You need to conserve, over here you need to conserve water. Okay, now when ANF is secreted, it is the opposite. Okay, it will allow diuresis to happen. Okay, water will be excreted out. Okay, but here the statement over here says decrease in secretion of renin by JG cells. Now renin, if you know the RAS statement, RAS hormone system, renin angiotensin aldosterone system. Okay, it plays a very important role in increasing the blood pressure or the blood volume in case it is low. Okay, so now renin forms angiotensin 1. I'm sorry, it actually is an enzyme which converts angiotensinogen to angiotensin 1, and then that is converted to angiotensin 2. Okay, now angiotensin 2 is a very powerful vasoconstrictor. Okay, it's a very powerful vasoconstrictor. Okay, so it will constrict the blood vessels. Okay, it will constrict the blood vessels. And this renin is secreted when there is low blood volume or blood pressure. Sorry. Okay, this renin is secreted when there is low blood volume or low blood pressure. Okay. Now our question is what? Which of the following would help in prevention of diuresis? But decrease in secretion will not help us. We need an increase in secretion of renin, right? When there's an increase in secretion of renin, yes, that will help in the prevention of diuresis. Now there are many functions of angiotensin 2. Okay, first of all, it acts on the adrenal cortex to release aldosterone. Also directly acts on the PCT, the DCT, the loop of Henle. It acts on them for sodium reabsorption to occur and water reabsorption to occur in those areas. And we'll come to that because there are many other factors. I don't want to go into detail now. Okay, so clearly your answer should have been B. Okay, the right answer should have been B. So to prevent diuresis, that is the renal output of water, you need reabsorption of sodium and water from renal tubules due to aldosterone. Okay, this was actually a good question. This is a very good question. So please pay attention, because now in the forthcoming slides I will be explaining how every hormone is responsible for reabsorption, or the opposite. Like in case of ANP, it is not reabsorption, right? It is allowing diuresis to happen. Okay, so if you understand this concept clearly, okay, you can answer any question, any damn question that comes with respect to this. You can answer it. Okay, so please pay attention. I would suggest you can you take down notes in your notebooks to help you understand. Okay, all right. So as per our question, yes, under secretion of ADH will reduce permeability of DCT and collecting tubules and ducts towards water. I've already explained that, right? So in the presence of ADH, it attaches to V2 receptors, and what does that do? Resulting in the increase of CMP and then activating the protein kinesis. So this is then intracellular reaction that's happening. And so as a result of that, this aquaporin 2, it's an intracellular protein, okay, and moves to the luminal side, okay, and fuses with the cell membrane by exocytosis and form water channels which are permeable to water. Okay, so water is allowed to enter through those aquaporin channels. Now, as I said, AQP3 and AQP4 are present in the basolateral side, which allow the exit of water molecules. Now, in the case of low ADH levels, AQP2, that's aquaporin 2, they move back to the cytoplasm and remove the water channels. Thus, they decrease the water permeability. Okay, so when there are low levels of ADH, there's no need for the aquaporin channels to be there, right? So they go back inside and they decrease the water permeability, and this happens in the DCT, collecting tubules and ducts. Okay, very important to remember. This is actually the later part of DCT. All right. Okay, so ADH, simple ADH also actually plays a very important role in determining whether you will be excreting a concentrated urine or a dilute urine. All right. So it's very important to understand the process of ADH. Okay. Aldosterone. Aldosterone is secreted by the zona glomerulosa of the adrenal cortex. Okay, they reabsorb sodium. So aldosterone plays an important role in reabsorbing sodium. Now the main action site of aldosterone is the principal cells of the cortical collection tubule. Also, yes, principal cells are also present in the later part of DCT. So there is some action also happening there. It doesn't mean that there's nothing happening there. Okay, so some action is also happening in the later part of DCT, apart from the cortical collection tubule. Alright, so change this. Alright, so the presence of sodium potassium ATPase pump causes the sodium to reabsorb into the blood and potassium to excrete. Okay, that is one of the functions. On top of that, it also makes the lumen side, the tubule lumen side, sodium permeable. Okay, so here you have the cell, here you have the lumen side, the tubule side, and you have the interstitial fluid side. Okay, so you have a pump over here. Okay, so now what we have to do, we have to reabsorb sodium, right? So anyway, the ATPase, the sodium potassium ATPase pump, what does it do? It kicks out 3 sodium ions and pulls in 2 potassium ions. On top of that, it makes this tubular side permeable to sodium ions. So hence this is the other way around. Hence sodium ions will move in. Make another one. Okay, so these are the two functions of aldosterone. Okay, so it activates this sodium potassium ATPase pump, and that creates a concentration gradient, and top of that it makes this tubular lumen side permeable to sodium ions. So that's how sodium ions get reabsorbed. Okay, now when sodium ions get reabsorbed, it increases the osmolarity of the ECF, the extracellular fluid. Okay, now when there are too many sodium ions, obviously the osmolarity of the extracellular fluid in your body will also increase. That's what happens when you consume lots of high sodium food. Also the same thing happens here. Sodium ion concentration increases in the blood, in the ECF also, and that increases the osmolarity. So what happens now, there are two ways by which water is absorbed. Now once sodium is reabsorbed, it increases its content in the ECF, that's extracellular fluid, and this increases the osmolality, and this increases the secretion of ADH. Now we saw what the function of ADH was, right? The function of ADH was to create those water channels, the aquaporin water channels, to reabsorb the water. Okay, water reabsorption should happen. Okay, so that is one part, and the other part is it activates the thirst center. Okay, so leading to consumption of more water, and this reduces the salt concentration to normal. Okay, this reduces the salt concentration to normal. So these are the two ways by which water, so water is not directly being reabsorbed, water is not by osmosis rather. Okay, so in the like I said, in the DCT, the later DCT part and the collection tubule, the cells are not permeable to water. Okay, so as by osmosis rather. So only when ADH comes and attaches itself over there, then that makes it permeable to water. Okay, so ADH helps in the reabsorption of water there, and also the thirst center is activated where you begin to when you physically consume more water. Okay, that's another way. And how is that aldosterone stimulated? With the help of angiotensin 2. Like I said, angiotensin 2 is a very powerful vasoconstrictor. Okay, it's a very powerful vasoconstrictor. It stimulates aldosterone for sodium reabsorption. Okay, so an increase in ANP released by the cardiac atrial muscle fiber is caused by the overstretch of the atria, which can be a result of high blood volume. Okay, so I previously mentioned why ANP is released, or why is ANP secreted. It detects a high blood volume. Okay, whenever there's a high blood volume or high blood pressure. Okay, so these cardiac atrial muscle, they sense that overstretch of the atria. Okay, they sense the overstretch of the atria, and because of that, that is due to a result of high blood volume, and because of that ANP is secreted. Okay, now what does the ANP do? It acts on the DCT collecting tubules and duct. Okay, and what it does is it causes a small increase in GFR because of vasodilation. Okay, so now obviously glomerular filtration rate, some glomerular filtration that happens at the glomerulus capsule. Okay, so vasodilation of the afferent arteriole happens. Okay, and that allows for more blood to be filtered. Okay, the more plasma is filtered. Okay, so that allows for small increases in GFR, causing vasodilation. As a result of that, there is decrease in sodium reabsorption. Okay, also it decreases the sodium reabsorption. Now what is our job? The point of this ANP is to decrease the blood volume. We already have a lot of high blood volume or high blood pressure. Our job is to decrease it. Okay, so what it does is it causes vasodilation. Okay, so that allows for more blood to be filtered. Okay, and it decreases the sodium reabsorption. Okay, in turn it decreases the sodium reabsorption, and so because of that diuresis happens. Okay, as a result of that there is diuresis. So water will also go out, right? So automatically the blood pressure, the blood volume decreases. So secretion of excess salt and water balances the excess blood volume. Okay, so this is actually the opposite of what aldosterone is trying to do, or ADH is trying to do. Okay, this is the opposite of that. So in aldosterone we are reabsorbing sodium, and because of that water is also being reabsorbed. Correct? In ADH also water reabsorption is happening to increase the blood volume, but this is the opposite. Okay, now we already have a lot of blood volume and the blood pressure is high. Okay, so this has to cause diuresis to happen. Okay, this has to cause diuresis to happen, and so because of this there are small increases in GFR causing vasodilation, and then this decreases the sodium reabsorption. Okay, all right, so I hope this was clear. Let's move on to the next one. All right, so renin. Now, renin is very important. Again, we saw the function of ADH, we saw the function of aldosterone, we saw the function of ANP, correct? Now, in which condition is aldosterone and ADH secreted? Okay, when you want to reabsorb more water and salt, correct? When there is low blood volume or low blood pressure. ANP is just the opposite. Okay. Now, renin is also secreted when there is low blood volume or low blood pressure. Is low blood volume or low blood pressure. And renin eventually forms angiotensin 2. Okay, renin eventually forms angiotensin 2. And angiotensin 2 is a very, very, very powerful vasoconstrictor. Please remember this, okay? Angiotensin 2 performs a lot of functions. It's a very powerful vasoconstrictor. So, renin converts angiotensinogen to angiotensin 1. Okay, it's an enzyme. Renin is an enzyme. Okay, it converts angiotensinogen to angiotensin 1, and angiotensin converting enzyme converts angiotensin 1 to angiotensin 2. Now, angiotensin 2 is a powerful sodium retaining hormone. Okay, I've already discussed that, and acts when there is low blood volume and low ECF. Okay, when there's low blood volume or low ECF, extracellular fluid, or low pressure, then angiotensin 2 plays a very, very, very important role. Okay, angiotensin 2 also plays an important role in activating aldosterone. Okay, come on, aldosterone, you got to do your job of reabsorbing sodium. Okay, so this is also one of its functions. Also, it directly acts on the all the PCT, the DCT, okay, on all those tubules. Okay, so it stimulates the secretion of aldosterone to reabsorb sodium. Okay, like I've already explained how aldosterone is working on the principal cells in the DCT and the collecting tubules, sodium potassium pump. Okay, so sodium is removed out, and also the luminal side, it makes the sodium more permeable. It makes the tubule luminal side more permeable to sodium. Okay, so this is one of the functions of angiotensin 2. Second, very interesting function it does is it constricts the efferent arteriole. Okay, in ANP we saw it vasodilates the afferent arteriole. Over here it constricts the efferent arteriole, okay, thus resulting in two effects. First one, it reduces the hydrostatic pressure, okay, in the peritubular capillaries, thus allowing for salt and water reabsorption. Okay, we'll come to that in a while, what I mean by that. And it increases the colloid osmotic pressure in the peritubular capillaries, thus increasing the reabsorption of sodium and water. Okay, so our job now is to increase the blood volume, correct? We need to increase the blood volume, we need to increase the salt and the water content in the electrolyte and the water content in our body. Okay, so it reduces the it constricts the efferent arteriole. Okay, the arteriole that is becoming into peritubular capillaries, it constricts that. Okay, so what happens? It reduces the hydrostatic pressure there. Okay, it reduces the hydrostatic pressure there, and this allows for salt and water reabsorption to happen. Okay, and in turn it increases the colloid osmotic pressure in the peritubular capillaries, thus increasing the reabsorption of sodium and water. Okay, we'll come to that. Yeah, so this diagram, this is taken from Guyton. Okay, he's written a textbook on medical physiology. Okay, so here, this is a normal condition on how reabsorption happens. Okay, this is a normal condition, okay, on how reabsorption happens. So now there are four forces working. Okay, there are two forces that are contributing to the reabsorption. There are two forces that are opposing the reabsorption. This is the hydrostatic pressure. of the peritubular capillaries. Clearly it is not allowing for reabsorption to happen. So it is opposing the reabsorption. This is the blood colloidal osmotic pressure. Okay, so this is promoting the reabsorption to happen. This is the hydrostatic pressure of the interstitial fluid. Yes, this is also allowing reabsorption to happen. So it is favoring reabsorption, and this is opposing the reabsorption. Okay, so we have understood that these four forces are acting. All right. So now imagine if the efferent arteriole is constricted. Okay, it will obviously, the hydrostatic pressure will reduce. Okay, it will allow for reabsorption now, because this is an opposing force. It now, if this is reduced, the hydrostatic pressure is reduced, it will allow for reabsorption of salt and water to happen. Correct? It will allow for reabsorption of salt and water to happen. Now, also another way that it does is, it increases the filtration fraction. It increases the filtration fraction. Okay, that is another job of what happens when there's constriction in the efferent arteriole. It increases also the of the angiotensin too. It increases the filtration fraction. That means more plasma is filtered. Okay, there is more than normal plasma that is filtered through the glomerulus. So what does that do? The efferent arteriole that contains that plasma, that blood that is going out, contains a concentration of salt. Okay, it contains a lot of concentration of salt, and that allows for water reabsorption to happen. That is how the blood colloid osmotic pressure increases. Okay, it allows for that reabsorption of sodium and water to happen. So these are the two ways by which angiotensin 2 acts. Okay, now once the efferent arteriole is constricted, these two things happen. Okay, these two functions happen. First is hydrostatic pressure decreases. First, obviously, we have seen it stimulates the secretion of aldosterone. On top of that, hydrostatic pressure decreases and the blood colloid osmotic pressure increases. Okay, by increasing the filtration fraction. Okay, so salt and water concentration increases in the peritubular capillaries, and that allows for blood colloid osmotic pressure to increase. Okay. Then, lastly, it directly stimulates sodium reabsorption, PCT, loop of Henle, DCT, and the collecting tubules. Okay, this is another third function of angiotensin 2. So, it stimulates the sodium potassium ATPase pump at the basolateral part of the cell, and then stimulates the sodium hydrogen exchange at the luminal side, especially in PCT. Okay, so the basolateral side, just write here, the base, I'm sure you're aware of this now, the ATPase pump. Sodium, it creates a sodium concentration gradient, right? So this is the counter transport mechanism. So sodium enters and hydrogen moves out. Okay, this is the luminal side, and this is the interstitial fluid side. Interstitial fluid side. Okay, the luminal side, where H plus ions are secreted out, and sodium ions enter. Okay, so the sodium potassium ATPase pump creates a concentration gradient over here. So right, so angiotensin 2, I have explained what are the functions of angiotensin 2. It stimulates aldosterone, it constricts the efferent arteriole, and directly acts on these tubules, the parts of the nephron. Okay, so this was clear. Now, the point of me explaining all these things, of all what ADH does, what ANP does, what aldosterone, what renin does, is for you to be able to understand that under what circumstances these are secreted and these are not secreted. Okay, because this is very important, because the subsequent questions that you will see, once you understand this logic behind every hormone and why it is being secreted or not, you will be able to answer all the other questions, right? Okay, so let's move on. Okay, so question number 2, let's see if we can answer this based on the information that we have. A decrease in blood pressure or volume will not cause the release of, okay, and this appeared in Neet 2017. Now, a decrease in blood pressure or volume will not cause the release of. Now, there's a decrease in blood pressure or volume. It will not cause the release of, obviously, atrial natriuretic factor or ANP. ANP. It's actually atrial natriuretic peptide. Now, an increase in blood volume or pressure or volume is will allow secretion of ANP. Okay, whenever there's an increase in pressure or volume, blood volume. Then aldosterone, yes, when there's a decrease in blood pressure or volume, yes, it will cause the release of aldosterone, it will cause the release of ADH, it will cause the release of renin. I've already explained the functions for you. See, now that is why I wanted you to go through that whole process. Okay, now there are 3 slides and 3 slides contain all the information. Okay, and I wanted you to be able to understand that. Okay, so the answer is A. See how Neet tricks you. Okay, now that was also a quite recent question, Neet 2000. That means that this is a very important topic for you to be able to understand how renin, how aldosterone, renin, aldo. Please go and read these functions, how ANP and how ADH, how they interact with the nephron and how they interact with each other. Okay, so this is a very, very, very important topic, in terms of Neet. Now you got another question. Okay, now a decrease in blood pressure or volume, decrease in blood pressure or volume will not allow the secretion of ANP. It's only when it's the opposite, when there's an increase in blood pressure or volume. Now we already know that, right? So clear with this? I guess I've already explained all the points related to this. So an decrease in blood pressure or volume, blood volume will not result in the secretion of ANP. On the other hand, it will cause the release of ADH, aldosterone and renin to reabsorb salt and water. Okay, so clear? Move on to the next one. Okay, this is another one related to what we have studied, right? Which of the following causes an increase in sodium reabsorption in the distal convoluted tubule? Now, which of the following causes an increase in sodium reabsorption? Increase in aldosterone levels, increase in ADH, decrease in aldosterone levels, decrease in ADH. And this appeared in 2014. We have just discussed this, right? So sodium reabsorption in DCT is increase in aldosterone levels. Okay, so answer is A. Clearly, aldosterone plays a role in the principal cells of DCT and cortical collection tubules. This is the main site, okay, for aldosterone to act on. Okay, and so when there's an increase in aldosterone levels, what happens? There's an increase in sodium reabsorption. Okay, and when is aldosterone secreted? It is activated by angiotensin 2. Okay, and when it is secreted? When there is low blood volume. Okay, when there's low blood volume, low blood pressure, that is when there is aldosterone secretion. Okay, which then acts on the DCT. Now you see how easy it is for you to answer those questions. So the late distal tube and cortical collection tube contain principal cells, okay, and they increase the sodium reabsorption. So when there is an increase in aldosterone levels. Oops, sorry. Went on to the next question. So to answer your question, let's go back. So which of the following causes an increase in sodium reabsorption? It is the aldosterone levels. Clear? This was again easy for you to understand if you knew that slide. Okay, if you knew those 3 slides, those 3 or 4 slides that are there, 4 slides I believe, please go back and study. Okay, because this is a very important topic, and how all these hormones are interacting with each other, it is very important. Okay, question number 4 now. So a fall in GFR activates. A fall in GFR activates. This appeared in mains 2012. Now why is there a fall in GFR? Okay, a fall in GFR will be a result of low blood pressure, right? There will be a low blood pressure. Okay, when there's a low blood pressure, there will be a fall in the GFR. So what happens during that? JG cells, the juxtaglomerular cells, to release renin, adrenal cortex to release aldosterone, adrenal medulla to release adrenaline, posterior pituitary to release vasopressin. Okay, so fall in glomerular filtration rate will immediately release renin. Okay, renin eventually forms angiotensin 2. Angiotensin 2 then activates aldosterone. See, they all interacting with one another, right? Now, aldosterone will act on the principal cells, then they will activate vasopressin. Okay, so do you get how they are all interacting with each other? Correct? So, a fall in GFR will activate the JG cells to release renin. So, your answer should be A. Okay, we'll go to the discussion. I think now I've made it very clear. Now you should be very used to this. A fall in GFR denotes low blood pressure. Okay, this stimulates the JG cells to release renin enzyme. So they eventually converts to angiotensin 2, which is a powerful vasoconstrictor. Okay, so clear? We'll move on to the next question. Okay. Question number 5. Which one of the following statements is correct with respect to kidney function regulation? Alright. A. When someone drinks lots of water, ADH release is suppressed. Exposure to cold temperature stimulates ADH release. An increase in glomerular blood flow stimulates formation of angiotensin 2. T. During summer, when body loses lots of water by evaporation, the release of ADH is suppressed, and this appeared in mains 2011. So clearly it is A, and we'll see why. So when someone drinks lots of water, ADH release is suppressed. Now I said ADH does to a certain extent determine whether we will excrete a concentrated urine or a dilute urine. Okay, so now when there is too much water, when it is a diluted urine, when there's too much water that needs to be excreted out, okay, when too much water needs to be excreted out, ADH levels will be suppressed. ADH secretion will be suppressed because we don't want water reabsorption to happen. We are actually excreting lots of water, so that results in the formation of dilute urine. Okay, so this is a very important function of the ADH. Now, when you are exposed to cold temperature, now in cold regions or whenever the temperature is cold, you don't sweat, you don't lose a lot of water. Okay, so that does not stimulate ADH release. Now ADH has to reabsorb water, right? That does not stimulate the ADH release. We want to remove the water. So clearly B is incorrect. Correct? C. An increase in glomerular blood flow stimulates formation of angiotensin 2. Clearly, we have just discussed in the previous question. A decrease in the glomerular blood flow. Okay, when there's a decrease in blood volume or blood pressure. Okay, it stimulates the formation of angiotensin 2. D. During summer, when body loses lots of water by evaporation. Correct. The release of ADH is suppressed. Clearly that is incorrect. We need ADH during that time. We need water reabsorption to happen. We want to conserve as much water as we can. We don't want to lose all that water. Correct? So that at that point, ADH then tries to form a concentrated urine by reabsorbing all the water so that we are able to conserve water. Obviously during summer we lose a lot of water by sweating, right? So our body then starts forming concentrated urine, and that is with the help of ADH. So ADH to a certain extent, yes, it does play an important role in determining concentrated or dilute urine. Okay, so this is a very important point. Okay, Clearly, then answer A is the correct answer. Okay, let's go on to the discussion. I think I've already discussed it mostly. So, in the case of excess water in the body, the plasma and the ECF osmolarity reduces, and hence the secretion of ADH is reduced. Okay. Therefore, water reabsorption rate becomes low in DCT and collection tube when large amounts of dilute urine is excreted. Now you know how ADH functions, right? So ADH to some extent, yes, very important point. Okay, to some this question actually wanted to test you whether you understand how ADH is important in determining whether dilute or a concentrated urine would be formed. All the options that were there were all pointing towards that particular topic. Okay, whether dilute or concentrated urine is excreted by the kidneys. So in cold temperature, obviously there is less sweating, hence ADH release is suppressed because water is conserved. So yeah, in case of angiotensin 2, a decrease in glomerular blood flow will stimulate the formation of angiotensin 2. And during summer, due to excessive sweating, it leads to increase in plasma and ECF osmolarity. So when excessive sweating and all happens, you're losing water, correct? So the plasma, so obviously the salt concentration in the plasma and the ECF will increase, right? So the body will try to conserve as much water as possible. That's why this ADH secretion is increased during that time. Okay, clear? Let's move on to the next question. Okay, so this is a very important table. I forgot to mention this, yes. A very important table, and it determines or denotes or shows the conditions where there is increased secretion of ADH and there is decreased secretion of ADH, and this was taken from Gyanam. He's written a book on medical physiology. So he has clearly denoted in tabular form the places where there is increased secretion of ADH and when there is decreased secretion of ADH. And this is very, very important. Very, very, please, very, very important. How ADH plays a very important role. Okay. So please note it down in your notebooks, on your notes, or wherever you want to. Okay. And we'll move on to the next question. Okay. So question number 6. Angiotensinogen is a protein produced and secreted by, you have options A, B, C, D. This appeared in 2006. JG cells, macula densa cells, endothelial cells, liver cells. So angiotensinogen is a protein that is produced and secreted by liver cells. So answer is D. Clear? This was direct. This you should have known. Okay, angiotensinogen is a protein that's produced from the liver cells. Okay, so answer is already there. It is synthesized in the liver and it contains 13% carbohydrates made up of 453 amino acid residues. Okay, it's extra piece of information. Main thing is you should remember that it is synthesized in the liver. Okay, so clear. Let's move on to the next question. Okay. If excess water passes out from the tissue without being restored by the kidneys, the cells would, and this appeared in 1994, burst open and die. So if excess water passes out from the tissue without being restored by the kidneys, so cells will not burst open and die. They take water from the plasma. No, that doesn't make sense. Obviously, the kidney is not restoring the water, so it can't take water from the plasma. Not be affected at all. No, there will be some change. D, shrivel and die. Yes, they will shrivel and die. So your answer should have been D. Okay, and we go to the explanation. So if excess water is passed out without being restored, what will happen? It will result in the increased osmolarity of ECF due to concentration of sodium. Okay, so when there's too much water that is being passed out, okay, this is a hypothetical situation that's given to us, and kidney is not able to restore that water. Okay, so there is obviously too much concentration of sodium now in our body, right, in the ECF. So the primary loss of water in ECF is called hyperosmotic dehydration. Okay, hyperosmotic dehydration. And also this happens in diabetes insipidus when there is inability to secrete ADH. Now you know what the function of ADH is, right? ADH reabsorbs the water through those water channels. Now if there's no ADH, imagine if there's no ADH, then water is continuously being excreted out, which is not a good thing, right? And this results in the hyperosmotic dehydration. Okay, so it creates a very hyperosmotic environment, okay, in the ECF, in the extracellular fluid, and this condition is called hyperosmotic dehydration. So when a hyperosmotic region or an environment is formed, what happens? To maintain that osmolarity, water from the tissues will move out, and they concentrating the intracellular fluid. Okay, so what happens if this cell is there? It's hyperosmosis. Water will move out. This is basic osmosis. I'm sure you're aware of this, right? When there's a hyperosmotic environment, okay, water will move out, and this concentrates the intracellular region, and they will all shrivel up and die. Okay, they all shrivel up and die. Okay, so remember, hyperosmotic region is also formed in the renal interstitial medulla, right? That allows for reabsorption of water to happen. Okay, that creates a hyperosmotic environment over there. So similarly over here, the primary loss of water in ECF is called hyperosmotic dehydration. Okay, and the kidneys are not able to restore the water. That happens in diabetes insipidus. Okay, when there's an inability to secrete ADH. Clear? So let's go back to our question. Yeah. So our question is, if excess water passes out from the tissue without being restored by the kidneys, the cells would shrivel and die. Okay, that will be the end of the cells. Okay, let's move on to the next question. Okay, so there's a match the following. Question number 8. Match the items given in column 1 with those in column 2 and select the correct option given below. This appeared in 2018 NEET exam. Okay, so you have column 1 and column 2. Ultrafiltration that happens in the Malpighian corpuscle. Concentration of urine occurs in Henle's loop. Transport of urine occurs in ureter. Storage of urine is in the urinary bladder. So what is our answer now? We have A4, B1, C2 and D3. So your answer should be B. Yeah, so your answer is B, right? So concentration of urine, Henle's loop is responsible. Ultrafiltration, Malpighian corpuscle. Transport of urine, ureter. Storage of urine, urinary bladder. Clearly, parts of the excretory system, you should be aware of that, right? Let's go on to the discussion. Okay, so you have your diagram here. This is from NCERT. Please study the diagrams of NCERT. So ultrafiltration occurs in Malpighian corpuscle. Urine concentration occurs in the loop of Henle. So it happens via countercurrent multiplication. Okay, that results in the hyperosmolarity of the renal interstitial fluid. This results in the conservation of water and the concentration of urine. I think we have discussed this in the previous. Please watch part 2. I have discussed this in a little bit of detail. So you can watch that. What are the conditions by which this urine concentration is determined. Okay. Ureter is responsible for transporting urine to the urinary bladder from the renal pelvis. Okay, you see the ureter over here. Okay, and the bladder, urinary bladder, is responsible for storing urine over here. Okay, clear? Let's move on to the next question. Okay, so question number 9. Human urine is usually acidic because this appeared in 2015. Potassium and sodium exchange generates acidity. No. Hydrogen ions are actively secreted into the filtrate. Yes. C. Sodium transporter exchanges one hydrogen ion for each sodium in the peritubular capillaries. Well, there is sodium and hydrogen counter transport mechanism, but does not happen in the peritubular capillaries. So C is incorrect. And D. Excreted plasma proteins are acidic. No, that's clearly incorrect. So the answer is B. Okay, let's go to the discussion. Now, human urine, as per NCRT, it is 6. The pH is 6. So it is slightly in the acidic region. Although a range, if you have to give, it's around 4.5 to 8. So it can vary between 4.5 and 8. Okay, so it can vary in this range. Although an average number would be pH is 6. Okay, just for your information, it is usually acidic. Now, to understand that H plus ions and HCO3 minus, that's bicarbonate ions, are secreted all the time. Okay, now our kidneys are designed in such a way that our kidneys are able to balance the acid and bases in our body. Okay, now in our body there are lots of metabolic reactions that are happening, right? So because of that also acids are formed, and acids need to be secreted out. Okay, so they are secreted out. Okay, along with that, when these acids are secreted out, there's a certain mechanism that is formed where bicarbonate ions are also reabsorbed. Okay, so for every H plus ion that is secreted, there's a bicarbonate ion that is being reabsorbed back into the blood. Okay, now there are two ways by which this happens. There's one way which is the sodium hydrogen pump. Okay, sodium hydrogen ion pump. I think if you watch my previous part, you will know what I'm talking about, right? So this acts in the PCT region, the thick ascending loop of Henle, and the first part, loop of Henle, and the first part, the initial part of DCT. Okay, so this counter transport mechanism, it reabsorbs sodium, it is excreting hydrogen ions also. It is secreting hydrogen ions also into the filtrate. It is reabsorbing sodium, yes, that's one of the job. It is also secreting hydrogen ions back into the filtrate. In turn, bicarbonate ions are produced. Now, whenever there's a secretion of H plus ions, bicarbonate ions are also produced, and this bicarbonate ions is being reabsorbed back into the blood. Okay, so we have to maintain a acid-base balance in our body at all times. Okay, so this is one contributing factor for the acidity, and this happens in the following regions. Now, in the later part of DCD and in the collecting tubules, and collecting tubules, there are special cells called intercalated cells. Okay, the special cells called intercalated cells. Okay, now these intercalated cells contain H plus ATP pump. They contain H plus ATP pump. Now this H plus ATP pump, it contributes only 5% of the secretion of the H plus ions into the filtrate. only 5, but this is so powerful that it will decrease the pH even more in the urine. That's compared to the sodium hydrogen pump. Okay, the sodium hydrogen ion pump is not really bringing down the pH as much as it should. Okay, it's present in a lot of places, but it is not contributing too much into the acidity as much as the H plus ATPase pump is contributing. All right, so this H plus ATPase pump plays an important role in bringing down the acidity of the urine even further. All right. So by these two mechanisms, we see the sodium hydrogen counter transport and the H plus ATPase pump, which is pumping out H plus ions. Okay, both are active transports, so hydrogens are actively secreted into the filtrate. This is a secondary active transport, and this is a primary active transport. Okay, so let's go on to the discussion. I've given you a brief. We'll go on to the discussion. Okay, so you have your diagram here. So when extracellular fluid contains H plus ions, they're regulated by the kidneys in two ways. I've already briefed you on that. There's secretion of H plus ions, and in return there's reabsorption of filtered bicarbonate ions. So bicarbonate ions are being present over here, are present in the filtrate. Okay, but there is a mechanism by which these bicarbonate ions get reabsorbed back into the blood. Okay, now let's see. Now we have the sodium potassium ATPase pump. We know how the sodium potassium ATPase pump works. Sodium is kicked out, potassium enters. It creates a concentration gradient. So sodium enters and this H plus ion gets secreted out into the filtrate. Now what happens inside the filtrate? This H plus ion combines with HCO3. Okay, now there is bicarbonate ions present inside. This H plus ions combines with the HCO3 minus, forms H2CO3, forms CO2 and water. Then comes the CO2 is able to go inside the cells. CO2 combines with water, forms H2CO3, again forms H plus and HCO3 minus. Again this H plus ion is used in the sodium hydrogen counter transport mechanism. Okay, and this bicarbonate ion that is formed here leaves. Okay, this bicarbonate ion formed here leaves. Okay, and this bicarbonate ion leaves via the sodium bicarbonate ion transport mechanism. Okay, there's a sodium bicarbonate ion pump over here, and bicarbonate ion goes back into the blood. This is one way. Okay, second way. This is the second way. Now there is H plus ATPase pump over here. So H plus is formed here. It is secreted out. So what happens? Actually CO2 enters, forms combines with water, forms H2CO3, forms bicarbonate. Okay, and H plus then gets secreted out. Here H plus, you notice, is not being recycled over here, unlike the previous case. Okay, here H plus is not being recycled over here like it is happening in the previous case. Okay, but instead a bicarbonate ion is formed, and this bicarbonate ions get exchanged with the chloride ions. Okay, so this is another way by which bicarbonate leaves the cells and goes into the interstitial fluid via the bicarbonate chloride exchange. Okay, and this happens where? In the DCT and in the collecting tubules. Okay, this happens in the DCT and the collecting tubules. This happens via the bicarbonate chloride exchange then. Okay, clear? So there are two ways. There's a sodium hydrogen exchange pump that is happening, right? Clear? And there's a H plus ATPase. So please remember, these two are the contributing factors to the acidity of the urine. Okay, they are contributing, they are secreting H plus ions into the lumen, into the filtrate. All right, so let's move on to the next question. And before I've taken this from Guyton, also familiar now. If you're watching my videos, you know who Guyton is. It's a very good textbook to study medical physiology, but don't worry about it in terms of NEET. When you study medicine, you will come across this book. Okay, let's move on to the next question. Okay, so question number 10. Which of the following does not favor the formation of large quantities of dilute urine? Okay, so does not favor. Let me just change this pen. So does not favor the formation of large quantities of dilute urine. Renin, does renin favor the formation of large quantities of dilute urine? Let's see. Renin is secreted when there is a low blood volume or blood pressure. Okay, low blood volume, blood pressure. Yes, it does not favor the formation of large quantities of dilute urine, right? Because renin tries to reabsorb sodium in water. So it is not favoring the formation of large quantities of dilute urine, right? Whereas atrial natriuretic factor, alcohol, caffeine, they are all diuretic. Okay, they favor the formation of dilute urine. They want ANP or ANF is secreted when there is a high blood volume or high blood pressure. Okay, so they want the water to be excreted out of the system. Okay, alcohol is a diuretic, caffeine is also a diuretic. Okay, so clearly it is renin. It does not favor the formation of large quantities of dilute urine. So answer is A. Okay, let's go to the discussion. Okay, so renin is secreted when there is a low blood volume or pressure, yes, resulting in excretion of concentrated urine. ANP, caffeine, alcohol, they favor the secretion of dilute urine. Okay, clear? All right, let's move on to the next question. All right, question number 11. What will happen if the stretch receptors of the urinary bladder wall are totally removed? And this appeared in 2009. Micturition will continue. Urine will continue to collect normally in the bladder. There will be no micturition. Urine will not collect in the bladder. Okay, so what will happen is the stretch receptors of the urinary bladder wall are totally removed. Now, stretch receptors play a role in the micturition reflex. Okay, that is the time when you want to go to the bathroom, right? So, if the stretch receptors are removed, what will happen? Then there will be no micturition. Okay, there will be no micturition. So now when these stretch receptors are present, so let's look at the answer. The answer is C. There will be no micturition. Okay, so let's go to the discussion. We'll discuss this in detail. So there are stretch receptors in the urinary bladder and they initiate the stretch reflex, and this begins the micturition contractions. Okay, so now what happens, the sensory signals are sent from the pelvic nerves and via the sacral segments and into the parasympathetic nerve fiber. So sensory signals are sent. Okay, and when the bladder is partially filled, okay, the contractions usually relax for a minute, then the detrusor muscles stop contracting and the pressure falls back. Okay, this is when partial full bladder is present. Okay, but as the bladder begins to fill up, okay, as the bladder begins to fill up, those reflexes become frequent. Okay, the reflexes become frequent, and then this causes greater contraction of detrusor muscle. Initially it is not so much when it's partially filled, but then the contractions become frequent. Okay, so the micturition reflex consists of, there's a rapid or progressive and rapid increase, okay, of the pressure, then sustained pressure period, and then return of the pressure, the basal tone. So as the bladder is getting filled up, okay, now let me just draw a better diagram, be able to explain it better. So just imagine if this bladder is getting filled up, okay, the volume is increasing. What happens to the micturition reflex? It will go up, sustain, come down. It will go up, sustain, come down. It will go up, sustain, come down. So what it means is return of the pressure, the basal tone of the bladder. That means the tone of the bladder, where the bladder is present, okay. Fill up, okay. Again, same thing happens, okay, and it continues to happen. So when the reflex is powerful enough, another reflex is sent. Now when there's too much pressure now, okay, when this reflex is very powerful, now it's become very far, the bladder is almost full, and now it's time for you to micturate, okay, during that time. So there are peduntal nerves, okay, which are attached to the external sphincter. The external sphincter is a voluntary muscle, correct? So a signal is sent there to inhibit that, okay. Now it's time to inhibit that, and so when that inhibits, micturiation will happen, and that is only possible if the signal that is sent to the brain is powerful enough, okay. If the signal that is sent to the brain is more powerful than the voluntary control, okay, when you want to sit and you want to micturate and it is powerful than the voluntary control of the muscle, micturiation will occur, okay. Micturiation will occur then. So if these stretch receptors were absent, clearly all this would not happen, okay. The sensory signals would not be sent. They would initiate the stretch reflex and then begin the micturiation contract. So all that would not occur. So clearly stretch receptors are responsible for micturiation reflex to start, okay. So micturiation reflex, like I said, there's a progressive increase, then the sustained pressure and decrease, a progressive increase, sustain, as the volume of the urine increases in the bladder, okay, as it is increasing the bladder. And when it becomes very strong, there's a signal sent to the peduntal nerves, to the external sphincter, okay, and that relaxes only when the signal sent from the brain is powerful enough, okay, and so micturiation will occur there. So to answer your question, let's go back to the question. So the what will happen is stretch receptors of the urinary bladder wall are totally removed. Then there will be no micturition. This is the appropriate answer for this. No micturiation will occur. Okay. Let's move on to the next question. All right. Yeah. So this is a diagram of the bladder. If you want, you can draw it in your book, or you may take notes of it, okay. So you can see the over here peduntal, and this is the parasympathetic, okay. Well, again, this is taken from Guyton. This is taken from Guyton, okay. So you can see the peduntal and the parasympathetic nerves here, and as well as the sympathetic, right, okay. Let's move on to the next question. Okay, so question number 12. A person who's on a long hunger strike and is surviving only on water will have A. Less amino acids in his urine, more glucose in his blood, less urea in his urine, more sodium in his urine. And this appeared in 2007. Well, so clearly less amino acids in his urine will not happen, because amino acids get converted to glucose by gluconeogenesis in starvation. More glucose in his blood, no, more glucose will not be present in his blood. Glucose is getting used up. So, but there will be less urea in his urine, and there will be less sodium in his urine, okay. I'm sorry, what is wrong with me? Okay, so question number 12. A person who's on a long hunger strike and is surviving only on water will have A. less amino acids in his urine, more glucose in his blood, less urea in his urine, more sodium in his urine. Well, clearly from the options we can say that a person who's on a long hunger strike, that means is undergoing starvation, will have less urea in his urine, and this appeared in 2007. Okay, we will look at the discussion. So the answer is C. The appropriate answer is less urea in his urine. Okay, so let's see what happens to our body in starvation. Okay, so you have the discussion here. So you can clearly see as the weeks of starvation increases, how carbohydrate, fat and protein reduces. Okay, we can clearly see that from the graph. So carbohydrate reserves will last for maybe half a day to a maximum of 1 day. Okay, so in here Guyton, as per Guyton, it says half a day, but in some other textbook it says 1 day, so it's an average. So carbohydrates will be utilized, okay, quickly. Similarly, now fats will start depleting. Fat stores will start depleting. Okay, so now there are two ways by which fat converts to glucose or ATP, okay, that you can use. Now triglycerides begin to move around more in the blood during starvation. Okay, and they break down to fatty acids, free fatty acids and glycerol. Okay, glycerol. So now this glycerol gets converted to glucose by gluconeogenesis, and these fatty acids then form acetyl CoA. Okay, they form acetyl CoA, which then enters the citric acid cycle. They then enter the citric acid cycle, and this citric acid cycle then form ATP. Okay, the citric acid cycle forms ATP. This is one way by which the fat produces energy. Another way by which the fat produces energy is that acetyl CoA breaks down into ketone bodies. Okay, so it breaks down into acetoacetic acid. So acetyl CoA breaks down into acetoacetic acetic acid. Acetic acid. Oh, this is one ketone body, keto acid. Okay, now that further breaks into beta hydroxybutyric acid, and some of it is converted to acetone and acetone. Some of it is converted to acetone. Okay, so these are ketone bodies. So now the body is going to ketosis. Okay, when these ketone bodies are formed, that means the body is going to ketosis. Now proteins play a very important role. Okay, so proteins, now utilization of proteins is happening by three ways. First is rapid depletion. That means there's formation of glucose via gluconeogenesis. That means amino acids get converted to glucose. Okay, this is during the early phase. So you can see the early phase. Okay, this is the early phase. There is some rapid depletion happening over here. Then there's a slow depletion. You can see this curve over here. Now when there's a slow depletion, that means that body has gone into ketosis. Okay, the body has gone into ketosis, where fat stores are being consumed quickly. During that period. Okay, so this is the slow depletion phase. Final rapid depletion. Now after that, again, when the fat stores are being consumed are over, now there is a rapid depletion phase over here. Which means that the protein, the intracellular, the stored proteins that are there in the cells, they are being used up for energy. Now when these proteins are being used up for energy, that means you are not going to survive any longer. Okay, this is the last phase. Okay, and eventually death occurs. Okay, eventually death will occur. Okay, once this final rapid depletion begins to happen. Okay, so stored proteins that are used for the cellular functions are being depleted. So this is the way carbohydrates, fats and proteins are being used up during starvation. Okay, so to go back to your question, clearly there will be less urea because the amino acids are being converted into glucose. Okay, and ketone bodies. And also another important interesting fact is once the glucose reserves are being used up, have already been used up, the keto bodies, the keto acids, they supply energy to the brain because they are the only other thing that can pass the blood brain barrier. Okay, they are the only other molecule apart from glucose that can pass the blood brain barrier and give energy to the brain. Okay, so this is another thing. So clearly there will be less urea in that person's urine. Okay, because amino acids have now been already utilized in the urea cycle. What deamination of amino acids happens because of that ammonia is formed, and with the urea cycle urea is formed, so there will be less urea in his urine. To answer your question, yeah. So clear. So in starvation you can note this down because it's very important. How carbohydrates, fats and proteins are utilized. Now with this information you can answer the next question. Okay, let's go to the next question. Question number 13. A person is undergoing prolonged fasting. His urine will be found to contain abnormal quantities of fats, amino acids, glucose, ketones. This appeared in 2005. Clearly it is ketones. I just explained it. Okay, the answer is D. Clearly it is ketones. Okay, so when the person is undergoing prolonged fasting, prolonged starvation, keto bodies will be formed. Okay, and these ketones provide energy to the brain. Okay, so this will appear in the urine, okay, in large quantities. Let's go to the discussion. So urine will contain abnormal quantities of ketones. Okay, and the acetoacetic, I've already told you, is formed from two molecules of acetyl CoA from fatty acids in the liver, and part of it is converted to beta hydroxybutyric acid. Minute quantities are converted to acetone. Okay, so these are the keto bodies. Okay, you can note this down. This will be helpful for you to understand to answer any questions related to starvation or anything like that. All right, okay, so clear. Let's move on to the next question. Okay, so question number 14. Use of an artificial kidney during hemodialysis may result in, this appeared in Neet 2019. Result in nitrogenous waste buildup in the body? No. That's not the purpose of artificial kidney. Non-elimination of excess potassium ions? No. There will be elimination of excess potassium ions. Reduced absorption of calcium ions from GIT, gastrointestinal tract? Yes. Reduced RBC production? Yes. So your answer should be D. Okay, answer D. Let's go to the discussion. So in the presence of artificial kidney, there will be no conversion of 25 hydroxycholecalciferol to 1,2,5 dihydroxycholecalciferol, which is an active form of vitamin D. Okay, this 1,2,5 dihydroxycholecalciferol is an active form of vitamin D. So if an artificial kidney is used, obviously this will not happen. So because of that, there will be less absorption of calcium in the intestinal epithelium, right? If this is absent over here, correct, there will be less absorption of calcium in the intestinal epithelium. Also, there will be no production of erythropoietin, which stimulates the production of RBCs. So this question focused on the other functions of kidney apart from the acid-base balance, osmoregularity, all that we have studied so far, right? But these are the other functions of the kidneys. Okay, they form this 1,2,5 dihydroxycalciferol, the active form of vitamin D, as well as erythropoietin. So it's important for you to understand the other functions of kidney, okay, for the exam. And this stimulates the production of RBCs. Okay, so let's move on to the next. Okay, so yeah, sorry. So urea, creatinine, phosphate, urate and sulfate are removed in large quantities from the uremic plasma in hemodialysis. So hemodialysis is an artificial kidney where a dialyzing fluid is placed there along with plasma, and so there is a net diffusion of components of molecules. So urea, creatinine, phosphate, urate and sulfate are removed in large quantities. Okay, next question. Okay, so it's a match the following. Match the items given in column 1 with those in column 2 and select the correct option given below. So we have column 1 and column 2. Let's match. Glycosuria. Glycosuria is presence of glucose in urine. Gout, accumulation of uric acid in joints. Renal calculi is mass of crystallized salts within the kidney. Glomerular nephritis is inflammation of glomeruli. So we have A4. Okay, yeah, we have A4, B1, C2 and D3. So answer is D. Answer is D, and this appeared in Neet 2018. Okay, so let's look at the discussion. Glycosuria is presence of excess glucose in urine. Okay, that means when transport maximum is limited or absent. Okay, so now obviously when transport maximum is the maximum amount of glucose that can be reabsorbed back into the blood. So if that is limited or is absent, then obviously there will be presence of excess glucose in the urine. Gout is caused by the deposition of monosodium urate monohydrate crystals, MSUM crystals at synovial joints. Okay, and it is formed from uric acid ions. And how is uric acid formed? As a result of metabolism of urine. So gout is actually the deposition of MSUM crystals, monosodium urate monohydrate crystals. So renal calculi or kidney stones is the precipitation of calcium phosphate crystals in the kidney forming calcium phosphate stones. Okay, so when this precipitation of calcium phosphate crystals, it forms calcium phosphate stones. Calcium oxalate stones are also formed, okay, as they cause calcium precipitation at high calcium levels. Okay, so calcium oxalate stones are also formed. Oops, sorry. Into the next question. So, and also the last one, glomerular nephritis is the inflammation of glomeruli. Okay, so that was easy to answer. Okay, we move on to the next question. Question number 16. A condition of failure of kidney to form urine is called, and this appeared in 1998. Condition of failure of kidney to form urine. Failure of kidney to form urine is called anuria. Simple, direct question, direct answer. Right? So answer is A. Deamination is the removal of the amino group. Okay, and uremia is the presence of high urea in the blood plasma. Okay, this high urea in blood plasma, and this happens in the case of renal failure. Okay, anuria also happens in the case of renal failure. Okay, in the kidneys or this renal failure, kidneys don't function like that, so then there is a failure of urine formation. Okay, so when there's a marked reduced renal blood flow to the glomerulus, it results in complete stop of urine output, and this condition is called as anuria. Okay, so there is no urine formation. Okay, so in severe cases of acute renal failure, anuria occurs and the patient can die in 18 to 14 days unless until there is an artificial kidney provided to it. So uremia is a condition of high concentration of urea in body fluids. Already discussed that. And deamination is the removal of amino groups from amino acids. Clear? The direct question, direct answer. Okay, next. Presence of RBC in urine is, this appeared in 1998. Presence of RBC in urine is hematuria. It's hematuria. Okay, the presence of RBCs in urine. So let's answer C. The others are clearly incorrect. We'll go to the discussion. So hematuria is the presence of RBCs in the urine. Urolithiasis. Actually, I think there has been a spelling mistake in the question, because there is no word as urothiasis. It's urolithiasis, and that is kidney stones. Urolithiasis is kidney stones. I think there has been a spelling mistake here. I think so. As per my research, there is no word as urothiasis, but if you know anything, let me know. It was urolithiasis as a presence of calcium phosphate stones formed in the kidneys. Proteinuria is the presence of excess levels of protein in the urine. And alkaptonuria is a rare genetic disorder. It's caused by the mutation of the gene for the enzyme homogentisate 1,2 dioxygenase. So when there is a metabolism of tyrosine, tyrosine and phenylalanine, okay, hemogentisic acid is formed. Okay, hemogentisic acid is formed. Now to metabolize homogentisic acid, homogentisate 1,2 dioxygenase enzyme is used for this metabolism, homogentisic acid. But now since there's a mutation of the HGD's gene, this enzyme is not produced. Okay, this enzyme is not produced, and because of that there will be a presence of this homogentisic acid in the urine. Okay, so now when this is excreted out into the urine, it forms an unusually black color. Okay, it forms an unusually black color, a dark in color. Okay, you can see your urine turning dark. Okay, now alkapton is also excreted in the urine. It's an oxidized form. Okay, that's why it's called alkaptonuria. Okay, alkapton is the oxidized form that is also secreted, okay, in the urine. So basically it is a presence of homogentisic acid, okay, and is formed from the metabolism of tyrosine and phenylalanine. Okay, and this causes the urine to turn unusually dark in color. Okay, clear? So I've covered this. If you go back to the question, so presence of RBC in urine is hematuria. Okay, clear? This hemat comes from the hemocytes. Yeah. So yeah, clear? Alright, so we've come to the end of this video lecture, and I hope you've enjoyed today's video lecture, and I hope you've learned something new today, which is very, very important. And please don't forget to like, share and subscribe to MJ Neet Biology. And I'm available on Facebook, Twitter and Instagram, so you can follow me there for more updates. The link will be there in the description box below. Okay, so don't forget, and I'll see you in the next video lecture. Alright, so bye.